Anyway, thank you very much, Eric. Just to introduce myself, I'm Cindy Davis. I'm a national program leader for human nutrition in the Agricultural Research Service part of the USDA. Today's session will focus on stage setting the current evidence base of precision nutrition, review what we already know, and identify the gaps in knowledge. Speakers will describe current research design and methodologies, limitations in design and data, and future challenges and opportunities for the field. We have 6 presentations, followed by panel discussion at the end of the session that will include all 6 speakers. Please type your questions into the chat box, and we will share them with the speakers during the Q&A. Because of the limited time for the session, I will not do long introductions for the speakers. Full bios for all of our speakers today can be found on the website. Our first speaker is John Mathers. Dr. Mathers is Professor of Human Nutrition, Director of the Human Nutrition Research Center, and Director of the Center for Healthier Lives at Newcastle University. Please join me in welcoming Dr. Mathers.
Cindy, thank you very much for that very warm welcome. It's a great pleasure to be part of this very interesting workshop. So in my presentation, I'm going to introduce some ideas about human variability and how those ideas might be used as a basis for developing personalized or precision nutrition. For the record, I have no conflicts of interest in relation to this presentation. So we're all different. We differ in obvious ways, such as how tall we are, or our eye color. We all have different body shapes, and we have different physical capabilities. We differ in how fast we grow and mature, and how rapidly we age. We also differ in how we respond to individual foods and diets. And to give you one example of that, I've taken some data from the Diets Fits study. This was a study of over 600 adults who took part in a 12-month weight loss trial. Now, the trial was compared a low-fat diet and a low-carbohydrate diet. And for the record, there was no difference overall between the 2 types of weight loss diet. But what's really interesting for us is the differences between the individuals and how they responded to being part of this weight loss trial. This waterfall plot shows you the individual weight changes for each of the 609 adults over the 12 months of the trial. If you look on the left of the screen, you'll see that some individuals did really well and lost 25 or more kilograms of body weight over the 12 months. As we move right across the screen, then you'll see that some individuals lost relatively little or no weight. And right at the end, on the right-hand side, you'll see that some people actually gained weight on this weight loss trial, sometimes as much as 10 kilograms in the 12 months. Another example of inter-individual differences, this time in response to supplements of fish oil. Fish oil have a number of biological actions, but one of them is in lowering triacylglycerol concentrations in blood. At least they do that in most people. In this study of over 300 individuals, we found a considerable number of individuals who didn't show any fall in triacylglycerols. These are the people who are described as non-responders on the plot. So these are people to the right of the vertical red line. So again, while some people show big falls in tag concentrations, some people showed no changes, and some people actually showed increases. So again, the take-home message is that we all respond differently to the same nutritional intervention. Now, one area which has been very fruitful as a way of exploring into individual variation is to look at the changes in blood glucose concentration following eating. This is relatively easy to measure. It's a low burden for the study participants, and changes in blood glucose after eating may be related to a number of health outcomes. And importantly, it differs between individual participants. To give you one example of this in action, I'm showing you some data from a study by Zevi and colleagues published a few years ago in Cell, where they used monitors to measure glucose concentration continuously over a number of days. And you can see in the top part of the slide the changes in glucose concentration over the day across a week. Now look at the bottom right-hand side, you see part of a day shown in detail. And using these plots, you can measure the postprandial glycemic response to either a standardized meal or to freely chosen meals. And what I want to focus on is the inter-individual variation in response to a standardized meal. And this is shown as the shaded blue area under the curve. And when that meal consisted of one of these four individual foods, either sugars, glucose, or fructose, or bread alone, or bread and butter, was a large inter-individual variation in glycemic responses. You can see this shown plotted as the frequency of an individual PPGR. Some people showing little or no response, some people showing big responses. And this has proved to be a very useful way of beginning to explore the metabolic responses to food. But what I'd like to do is to take that into the public health arena and to think about how we might use this information on inter-individual variation to improve public health. This is because diet really matters when it comes to health. I'm showing you here some data from the Global Burden of Disease Consortium, showing you the contribution of different features of diet to morbidity and mortality globally. Whether you live in a high SDI country or a low middle SDI country, what we eat really matters in terms of our health. So the public health challenge is to improve everyone's eating habits so that eventually we improve public health. The current public health approaches to changing diet are relatively ineffective. In most cases, it involves giving everyone more or less the same advice. For example, to consume at least 5 portions of fruits and vegetables per day. What we'd like to know is whether taking a precision or personalized nutrition approach might do better. In other words, if we put the individual in the center and think about what really matters for them, could we develop nutritional advice and support which enables that individual to improve their diet more than they could do when they're offered the one-size-fits-all type of advice. So let's think a little bit about what personalized nutrition and precision nutrition means. And here I'm giving you some examples of definitions that we used in a paper we published with Jose Ortavas a few years ago. Now here we thought of personalized nutrition as an approach that uses information on individual characteristics to develop targeted nutritional advice, products, or services. On the other hand, precision nutrition is more ambitious. It suggests that it's possible to have a sufficient quantitative understanding about those complex relationships between an individual, his or her food consumption, his or her phenotype, including health, that we could offer nutritional intervention or advice which is known to be individually beneficial. Precision nutrition demands much greater scientific certainty. So that then leads to a number of questions. One of those is, what is the basis, the biological basis for this individual variation? And I've given you some examples already. And secondly, can we use knowledge of that basis of individual variation to develop more effective, personalized or precision nutrition. So it's certainly true that genotype matters and that we might be able to use information on our genetic differences, so the individual changes in the sequence of our DNA, because that changes the way in which we respond to food and may impact on our health. The genotype's not the only biological feature which matters. Other relevant biological factors include the epigenome, set of marks and molecules which regulate DNA and ensure that we express the appropriate genes in the appropriate cells under the appropriate conditions. We're learning too that our gut microbiome also matters, and that may influence how we respond to foods and indeed how that influences our long-term health. And we'll hear more about these areas throughout this workshop. But it's not just biology, and we also need to include psychology. I'm going to show you an example from one particular study which illustrates the complexity of this relationship between psychology and biology. And this was a study in which a group of individuals underwent the same measurements on 2 occasions. In this study, on the first occasion, the individuals consumed a meal, and the researchers measured responses in terms of satiety and subjective experience of the participants. Then the individuals were informed about their genotype. Now, the clever thing about this study was that what the researchers did was to randomize the genotypes so that people were told a genotype at random, not their actual genotype, but in such a way that people were balanced, so that some people were told they had the high-risk genotype and some people had the protective genotype. And then the measurements were repeated. They consumed the test meal and the same measurements were made. In this case, the genotype was in relation to the gene FTO, and this is the gene which has the single biggest effect on obesity, on the risk of obesity in the general population. So on the right-hand side, you'll see the responses of the individuals. On the left, in red, are the responses of those individuals who were told they carried the high-risk version of FTO. And in blue on the right, the responses of the individuals who were told they carried the protective genotype. And what we're looking at are 2 aspects of response. And we're comparing the measurements on week 1 before they knew about their genotype and week 2 after being told about their genotype. And what you see is that for those individuals who were told they had the high-risk allele, they showed no difference in response between the 2 weeks. However, for those individuals who were told they carried the protective allele, they showed a much bigger response in the hormone GLP-1 and also a much bigger self-reported fullness measure of satiety. So this suggests that learning one's genetic risk changes our physiology independent of actual genetic risk. Now, this is a fairly recent study and it needs to be repeated. But if this is true, then we need to think carefully about the implications of using genetic information in the design of precision or personalized nutrition interventions. So I'm going to explore a little bit further the basis for inter-individual variation in response to diet. And I want to think about timescales, because what matters when we're thinking about changes over hours or minutes is very different from what matters when we're thinking about weeks, months, or years. To illustrate that, I'm going to go back to the example of looking at changes in blood glucose concentration after consuming food. So if we look at the glycemic response in the first 2 or 3 hours after consuming a meal, almost all of that inter-individual biological variation is probably down to the biology of the consumers, with much smaller contributions from psychology and social factors. However, if we're thinking about long-term responses to food, responses over months and years that might lead eventually, for example, to insulin resistance or to the diagnosis of diabetes, then it's likely that we'll see a reversal of the importance of these factors, and that social factors and psychological factors may become at least, if not more important than biological factors, because these determine our eating behavior in the long term. So the contributions to health change when we think about timescales. So when we're thinking about precision and personalized nutrition, and we're thinking about the causes of inter-individual variation, then I think we should be looking at this bigger picture and thinking about a biopsychosocial model. So let me now turn to translation of these ideas into the development of precision and personalized nutrition. If we're thinking of this from a public health perspective, then we need to ask questions such as, why should individuals engage with precision or personalized nutrition? Does this really fit in with their aspirations? We all have different aims and ambitions. For some of us, it might focus around what we look like or our performance in sports. We have different likes and dislikes. And we have different food preferences. And all of these factors need to be considered if we're thinking about changing eating behaviors using a precision or personalized nutrition approach. If we think about this over the longer term, then we need to think about the individual barriers and facilitators for dietary change. And these will differ between individuals. And one of the challenges for personalized and precision nutrition is being able to identify and measure these barriers and facilitators and include those in the personalized nutrition or precision nutrition offering. The final area I want to discuss is inequity in diet and health. I'm going to start with an example from the UK where I live and work. And this shows you the prevalence of obesity in children at 2 ages. In red at age 4 and at age 10 in blue. And what you see looking from left to right is that across the socioeconomic profile between the richest families and the poorest families, there's a twofold difference in risk of obesity, with the children from the poorer families having twice the risk of obesity as the children from the richer families. And that's true when the children are 4 years old, and it continues to be true when they're 10 years of age. Now an example from the USA. The USA, like many countries, has been experiencing an increase in life expectancy in general, which has been going on for decades. However, after the year 2000, the poorest in the USA have not seen an increase in life expectancy in the way the rest of the population has. So there's inequity in life expectancy and other aspects of health, which is linked to socioeconomic differences. When we're thinking about personalized and precision nutrition, we should also be thinking about addressing inequity and improving opportunity for everyone. summarizing some of the challenges for precision and personalized nutrition, I think you can consider these in two groups, those which are important for the individual and those that are important for society. So for the individual, personalized or precision nutrition should be more accessible than current approaches, more attractive and more acceptable, should be more motivating and create more opportunities. And again, that will help to address some of those issues of inequity. For society, precision and personalized nutrition should have greater reach, should be more affordable and more cost effective, and importantly, should be sustainable. That is, it should have set up systems that sustain behavior change in the long term. Existing evidence shows that personalized nutrition approaches can improve dietary intake, and we saw this in the Food for Me study that I was involved in that was carried across 7 European countries and showed that personalized nutrition improved dietary intake compared with a control one-size-fits-all approach. And then in a more recent review of 11 studies, overall, personalized nutrition approaches improve dietary intake in healthy adults. We may be able to improve the effectiveness of precision and personalized nutrition if we take a systems approach. So if we have a comprehensive way of quantifying relevant participant characteristics, including measuring those individual barriers and facilitators to dietary change, and working with individuals' aspirations to develop personalized or precision nutrition which really works for the individual. Then linking that with a self-monitoring process which feeds back into the participant characteristics and round the cycle improving the personalized or precision nutrition and eventually leading to increased health and well-being. So to summarize, we all differ in many ways. Genetic factors explain some of those inter-individual differences, but our epigenome and our microbiome are also important. As we develop more effective precision and personalized nutrition strategies, we should not just think about biology, but also think about psychology and sociological factors. The future developments of precision and personalized nutrition should include greater focus on individual aspirations, on individual barriers and facilitators, and they should be set up to address inequity. Thank you very much.
Thank you, Dr. Mathers, for that wonderful presentation to set the stage about the many different variables and challenges that need to be considered for a precision nutrition approach.